I would like to thank Gordon first to invite me to beautiful Split. I'm not going to show a lot of mass spec data, so we're going to shift a little bit gears. And I would like to talk about some of our work, and this part is mainly focusing on how H3 and 2 viruses evolve their receptor binding specificity according to antigenic pressure of our immune system. And I don't think I have to tell you guys how complex this glycocalyx is. You guys really love your N-glycans, and I do too. So if we look at glycome binding by viruses, we like to consider that these are very highly evolved. These viruses are with us and other species for a long time, right? And during our studies, we have shown, for example, that influenza viruses can bind N-glycans in a bidentate manner. We see different viruses, noroviruses, binding blood group antigens. So some people do not get noroviruses because of the blood groups. And so we have a lot of different viruses, enveloped capsid viruses, with highly evolved glycan specificities that we, in turn, have select receptor and cell tropism, because all these glycans are differentially displayed on different cells, organs between species, and therefore you have viruses that can cross species, but you also have a lot of viruses that bind glycans that are restricted to a certain species. And if we look at influenza viruses, we consider this sialic acid linkage, I made the linkage bigger, as the host species barrier. And when we look at avian viruses, these bind to sialic acids linked to this third carbon of a galactose, the chemical structures here and the symbol nomenclature. And then you have human viruses that bind to this exact same sialic acid, but now linked to the sixth carbon. And what we know about these linkages is that they're differentially displayed in your respiratory tract between species. So whereas humans display a lot of alpha-2,6 linked structures in their nose, resulting in efficient transmission of these viruses, right? You sneeze. We see, and that's actually interesting in our favorite animal model of the ferret, we see a very similar distribution of alpha-2,3, alpha-2,6. But for example, in chickens, we see mainly 2,3 in their respiratory tract and gastrointestinal tract. And in pigs, we also see a lot of 2,6, but those are mostly N-glycolyl sialic acid and not nu-X structures. And human influenza viruses do not bind N-glycolyl. Okay, so very important here, if you have 2,3 specificity, you have the sialic acid here in the receptor binding site, 2,3 specificity, you have this gal, Gluckneck, and that goes out of the receptor binding site. For 2,6, this is remarkably different, because you have this 2,6 torsion angle, and now this Gluckneck structure goes over additional structural features of a human HA, making additional interactions there. And this is the specificity, right? It's not the sialic acid, because that's exactly the same in these receptor binding domains. It's the linkage of sialic acid. And this is a study from now more than 10 years ago, where some of your colleagues showed that you have a bunch of very complex N-glycans in our upper respiratory tract, which is mainly illustrated by this enormous amount of Gluckneck repeats. And during my studies, also back in San Diego, we made these structures, right? We do chemoenzymatic synthesis and we make these structures. And a lot of research eventually pointed out that we see this bidentate binding event if this structure has 3 Gluckneck repeats, 1, 2, 3. And actually for an H7 influenza virus, we were able to turn this on and off with a single amino acid, right? If we have a lysine here, we push the Gluckneck out, and there's a threonine, there's space, and now this HA was able to bind this structure. So we make these structures, and a thing that we do in the lab is we print glycan arrays. So this is a pin that goes into a 96 well plate, where it's a defined glycan, and it deposits on 6 replicates on a glass slide. And we know exactly where these structures are printed on these slides. Actually, I always have to tell people that Geert-Jan is right here. And when we have these glycan arrays, we can analyze these either with whole viruses, and that's what I'm going to mainly talk about, but we also have a lot of approaches to create recombinant proteins, which we make multivalent with antibodies. And then we probe these glycan arrays and can assess receptor specificity of not only influenza viruses, but many others and proteins. And one of the problems in influenza virus research is that this H3N2 virus is outsmarting us. And which is actually odd, because this virus has been in the human population since 1968. And you can see a phylogenetic tree here, and every different color is a different vaccine strain. Right? Every other year we make antibodies, these viruses escape antibodies, and there are quite some, 1, 2, 3, 4, 5, like 10. It stops at 2002, because after 2002 we had 27 vaccine updates. And the reason is that these viruses have a declining receptor binding avidity. They don't bind to erythrocytes anymore, and they actually also grow very poorly in our substrates. And this has resulted in extremely low vaccine efficiency. For H3N2, some years we were looking at 20–30%. So it only works in 1 out of 3 people. And the main reason here is this lack of binding to erythrocytes. And we really love erythrocytes. And this is how we look at this. We have red blood cells. You put in a 96 well plate. What happens? These red blood cells fall down, right? And you get a dot. On the moment you add a virus, with an hemagglutinin, hence the name hemagglutinin, you get a smear of red blood cells. And very beautifully, you can add titrate serum in this assay, and this serum will inhibit hemagglutination. And if you can titrate dilute serum 40 times, it's a correlate of protection determined by the WHO as being protective. On the other hand, if you have viruses that don't bind red blood cells, you don't have anything to inhibit, and you cannot perform this assay. So we took an approach here with 2 PhD students here, Rosanne and Frederik, and they took a bunch of H3N2 viruses with our collaborators in Rotterdam. These are all isolated in the Netherlands, and we have 91, 2003, 2009, 17, and 19. And hopefully you can see that on the moment we are in 2009, and here these viruses do not bind any erythrocyte anymore. You have a couple of viruses that regain this ability, but these are antigenically different viruses. We actually have 2 antigenic specific H3N2 circulating in the population. But this was a major problem, right? You cannot perform antigenic analysis anymore if these viruses do not bind erythrocytes. So we tested these viruses on our glycan arrays, and so we have symmetrical structures, but we really like to make asymmetrical structures, as hardly anything in nature is symmetrical. And so we made them with 2 and 3 leg-neck repeats. SNA binds all to 6. In a 91, you can see that these viruses bind all kinds of structures, also the very simple one. And already in 2003, you can hopefully appreciate this decline in receptor binding breadth, right? These viruses now do not bind these simple structures anymore, but only structure 14 and 17 with these tri-leg-neck structures. And that's what we saw for 2009, 17, 19. So these are the different antigenic clades. And then 3C3 clades were regaining some di-leg-neck binding. But they still bind very poorly to erythrocytes. And so we were able to make these structures in a test tube, and we wondered, can we make these structures in the same test tube, but then with erythrocytes? So we actually took erythrocytes, released sialic acid with neuraminidase, and then added B3GNT2, B4GALT1 to build up these leg-neck structures. And then after a wash, you can really wash erythrocytes quite easily and add 2 6-link sialic acids. And then we look at their receptor binding of the same viruses. So these 91 viruses, they don't mind, right? They just bind wild-type erythrocytes. 2003, the same thing. but on the moment we're in 2009, it doesn't bind unmodified erythrocytes anymore, just to 6 only polylacnac or to 6 polylacnac, and in 2017-19 only these modified red blood cells are bound by these viruses. And we saw this for many different viruses that were circulating at the time, and we still do this for antigenic surveillance in the Netherlands, but also for the UK. So we took a whole set of viruses just to make our point from 1968 right on 2019, and just to make a long story short, these viruses hardly bind chicken or turkey erythrocytes if they're unmodified or just recellulated, but they all bind pretty high titers to our modified erythrocytes. And that's great because now we can do antigenic characterization of these viruses again, right? We can now inhibit this hemagglutination profiles. And that's exactly what we did. This is what virologists look like. They take a virus, they take serum. These are vaccine strains of different years. They come here as well. So in bold, you have the homologous titers, and we can actually see that that looks very well, because same virus, same serum. But on the other hand, if you have different antigenic clade, different vaccine strains, the protection is very poorly. And you can also put this in a grid. So every grid is a different dilution factor. So as an example, this was a 3C3 virus vaccine strain in 2019. The 3C3s that circulated were actually already quite antigenically distant, but the majority of the viruses, this was 90% of the viruses that were circulating, were from the 3C2A1 clade. If you got the vaccine, you were not protected against these viruses. And that was a major problem just before the pandemic. And so we create glycoengineered red blood cells to enable this antigenic characterization. And we also, of course, analyze these red blood cells with mass spec. And I'm really glad we first did the phenotypic assays, because we only found like 1% abundance of these very complex N-glycans. So you only need a couple of N-glycans that are the preferred receptors to get high efficient hemagglutination. We also figured out that we only need one branch of alpha-2,6 trilacnac as a minimal receptor. We developed here glycan array analysis with whole viruses. We just take supernatants and detect them with broadly neutralizing antibodies. And one of the ideas we had is to increase these structures in MDCK cells to get higher titers of virus or isolate viruses that we could never do because these viruses could not infect these cells. So we overexpressed B3GNT2 in these cells, which were already overexpressing alpha-2,6, and we were able to make beautiful HCK cells with very extended N-glycans. But it never led to higher titers or for better isolation. So you only need a couple of glycans to get infection. And so we were wondering, is this the end of the story? Not really, because these viruses continuously evolve, especially after the pandemic, 2002, 2003, H3N2 viruses came back into the population. So what we were wondering on one moment, is there an arm preference? And you already seen, we only have one ST6L1 enzyme, and that actually has a preference for a distinct arm. It really prefers the alpha-3 arm. And we were wondering what happens next. And actually, we discovered that these viruses revert to dilacnac binding. So we made this little glycan array, all with asymmetrical structures, and they have elongation on alpha-6, or they have an elongation on the alpha-3 arm. And then we tested all our viruses on this array again. So you can see in 9068 binds everything that's alpha-2,6, and you can see this evolution again. And here in 2016-9, you can still see it binds some dilacnac structures, right in these, but in a lot of other viruses don't. And then in 2021, 22, we actually saw this phenotype coming back. And I would really like to emphasize that these are structures Q and O. So O and Q, whereas P and R are not bound. And the difference is the elongation of the alpha-3 arm instead of the alpha-6 arm. So these viruses bind to the alpha-3 arm, which makes sense because ST6-cal preferentially CLLates this arm. And next we were wondering, what are the molecular features? How are these viruses evolving? And this has been work of Ryonan, who's still in the laboratory, which was 2025, sorry, it's just out. And then she was wondering, how do these viruses bind to dilacnac or trilacnac? And she took a couple of specific viruses, Switzerland NSK 158, 160, and a Singapore 2016 virus, NYT. And one of the things that we were wondering, because a glycan array is so much, right? It's a screen. You detect binding, but it's a qualitative output. If there's a little peak and a big peak, we just don't know if that actually is an avidity difference. So, and we made these structures in the lab so we can also biotinylate these structures and actually employ them in ELISA format. So we get an idea about the relative avidities. That's what we did. So we took a small set of glycans, 1 lacnac, 2 lacnac. Of course, we focused on the preferred arm at the alpha-2-3, and then we did ELISAs. And what we mainly saw is that this is actually an N-glycosylation site. So if you introduce this N-glycosylation site, NST, in the Swiss, you can see it loses a lot of binding. It only binds to this symmetrical trilacnate containing N-glycan. And we see that it really, all these viruses bind these structures. And very importantly is this Y that's very important for binding these dilacnac structures. And that's what we see here, because on one moment we observed that in 2022 we went from NYT to NNY at this position. And this position is really antigenic. On the moment we see mutations here, we know we're going to go into an antigenic clade and we have to update our vaccine strains. So this is the Singapore wild type, and in the moment we mutate out this tyrosine at 159, boom, it does not bind anything anymore. And in other studies we've shown that this tyrosine is absolutely essential for proper receptor binding, although it's very far away from the receptor binding site. Actually, we've shown with NMR that this tyrosine directly interacts with the glucnac-6. And on the moment we have this NNY, we see very poor binding. It still binds to this asymmetrical trilacnate containing structure, but not to this asymmetrical structure. And what Ryonan did, she did tissue binding. We have ferret lung tissue and human tracheas. So this N that doesn't bind anything, of course, doesn't bind tissues. But I would really like to emphasize that this HA that does bind these trilacnate symmetrical structures does not bind to tissues. Does not bind. It binds this very complex glycan, but it does not bind tissues. And what then Ryonan did, she went into the whole evolution. I think it was a major effort of her because all of other amino acid positions also change in evolution. And maybe I don't have the time to go into the exact data, but what I really would like to emphasize is that on one moment we found this triple mutant. This is Y95F, and we can see that it really binds very strongly to these dilacnate structures. Everything in the NNI background had problematic binding avidity, but it was all rescued by this single I95F. And that tyrosine 95 is actually 100% conserved in all influenza viruses. So we were very surprised that we would find this. And she did all kinds of other 777 mutants. And this is the Darwin strain. This is the vaccine strain that we had until last year. And you can see that this vaccine strain actually now binds dilacnate structures as well. And she did the tissue, first the glycan array analysis. So if you see Dilag neck binding in the ELISA, you see dilag neck binding in the array with these structures. And if you don't see any of dilag neck binding, you really don't see this back in the array. And if we then look at tissue binding, the thing that I really would like to emphasize, on the moment you only bind trilag neck containing N-glycans, you don't bind tissues. And this is the Darwin strain, which very efficiently binds ferrets, lungs, and human tracheas. So we actually also wanted to, if you only bind this symmetrical trilag containing N-glycan, it's not present on tissues. So human influenza viruses continuously evolve their receptor binding properties. Eventually they have pretty similar specificities with dilag neck structures. And these specificities can be achieved with different receptor binding site constellation. And we see actually every couple of years this binding dilag neck, losing dilag neck, and it depends on the antigenic clades. This single trilag neck arm is is probably displayed in ferret and human respiratory tract, so only these asymmetrical structures, not the symmetrical structure. And we've seen that several of these specificities can be very restrictive for these viruses. And with that, I would like to thank the previous and past and present members of the laboratory. I have to thank, of course, my collaborators at Erasmus who do all these antigenic analysis. We still make these erythrocytes. It was actually very important last year again, because these viruses were really not binding erythrocytes. And we had another vaccine strain. Just as a fun side note, it's the vaccine strain for the Northern Hemisphere next season is Croatia. It's a Croatian strain. We were very disappointed in the Netherlands because we also put it forward in a Dutch vaccine strain, and the WHO recommended the Croatian one. FYI. We make these erythrocytes exactly for this. And I have to thank, of course, also the collaborators from Spain and the University of Georgia. And with that, I would also like to thank you for your attention.